Hi everyone, my name is Claire O'Connell and I'm from the School of Physical Sciences. My talk today is going to be Where's Wally and the search for cancer cells. So cancer, as I'm sure you're all well aware, is a leading cause of death worldwide. But what some people do not know is that these deaths are caused by metastasis. This happens when the cancer spreads to the rest of your body. So let's just talk about that for a second. So say you have a primary cancer tumor somewhere in your body. Cells from that tumor can break off and enter your bloodstream. If they can get into your bloodstream, they have access to every one of your vital organs. And obviously, if the cancer spreads, it makes it much more difficult to treat. The idea for my PhD is to be able to find these cancer cells in your bloodstream in low numbers in order to detect the cancer earlier. The challenge with this is that there are about 1 to 100 of these cancer cells among a billion healthy blood cells. So it is the proverbial needle in a haystack situation. But one of the nice things about this work is that it can be applied to many types of cancer. In fact, it can be applied to any cancer that metastasizes, which is the vast majority. But it includes lung cancer, pancreatic, colorectal, breast, or prostate cancer. But for the rest of this talk, I'm just going to talk about the work I've done on prostate cancer. So how do I propose to find these cancer cells in your bloodstream? Well, I'm going to get my good friend Wally here to help me explain. So some of you will be more familiar with him in images such as one here. So quick hands up. Who can find him in the picture right there now? No one? Okay, I didn't think so. But what if I gave him a pair of glasses that lit up? Then if I turn off the lights, we'd literally be able to find him in a number of seconds. This is the exact same logic I want to apply for my PhD. I want to tag the cancer cells with little lights in order to distinguish them from your healthy blood cells. So how am I going to make these little lights? Well, first I'm going to take a special dye. And it's special because it fluoresces, which means that it emits light when we shine red light on them. But I'm not just going to take one of these dyes. I'm going to take hundreds of them. And I'm also going to put them inside a silica shell, which is just another word for glass. So essentially, I'm putting the dye molecules inside a glass case to keep them all together. So these particles are about 100 nanometers in size, which is why we call them nanoparticles. They're about 1,000 times smaller than the width of a human hair. So they're really quite small. But they also can't distinguish between cancer cells and blood cells at the moment. So that's where antibodies come in. These are structures that can target specific proteins. And cancer cells actually have different proteins on their surface than your normal healthy blood ones. So we can program these antibodies to target the proteins on the surface of the cancer cells. This way, when we put the nanoparticles in with the mixed cell population, they should only adhere to the cancer cells. So this is the theory behind how my project should work. So if I want to mimic a patient's blood sample in the lab, I'll first have to take some blood. But let's just look at it for a moment. So it's comprised of 4 parts: plasma, which is the fluid, red blood cells, white blood cells, and platelets. Now, if you had these circulating tumor cells in your blood, they would end up in the white blood cell and platelet fraction of your blood. So what I'll do is I'll ask a donor who's healthy to give me their white blood cells and platelets. But I will count the number of white blood cells that are in there beforehand. And I will spike in some cancer cells, some prostate ones that were cultured in the lab previously, and I'll spike them in at different ratios. So if you had early stage cancer, you would be able to find these cancer cells at roughly 1,000 to 1 white blood cells to cancer cells. So I will spike those cancer cells in up to this ratio to mimic a patient's sample. So lastly then, the magic ingredient, I add the nanoparticles in and I let them incubate together for a while. So now I need to figure out if the nanoparticles have tagged the cancer cells correctly. So what I'll do is I'll bring it to an instrument that can do this, a flow cytometer. So what this does is it draws cells one at a time through a laser beam, and then it can detect the amount of light that is emitted from each cell. So say if it was a prostate cancer cell that was in your blood, you would see something like this, where the nanoparticles have directly attached to the surface. But if it was a healthy blood cell, we wouldn't see anything out here at all. So far, I've done a number of tests where I've spiked in the prostate cells with white blood cells, and I've done this at different ratios. And I've been successful in finding those cancer cells at 10 to 1, 50 to 1, 100 to 1, but not quite at 1,000 to 1 just yet. So I'm currently trying to push the limit of detection to this 1,000 to 1 clinically relevant range so that I can help develop a test that will be able to detect early cancer at an earlier stage. Thanks for listening.